Hello, my name is Sperling. I work in ARO in Israel, and I work with Ranjit Karan to study what we call P2B, and that is to determine the role of phosphorus in promoting almond yields. Um, a bit of general background. So P supports plant physiology. Nearly 0.2% of most plant tissues there is almost 0.2% of phosphorus in all plant tissues, and it supports photosynthesis, respiration, protein formation, membrane structure, enzyme activation, and multiple processes that require biological signaling. It makes P critical for plants and also for farming, because essentially crops cannot access almost 80% of the soil P. P moves very slowly by diffusion in soil, and P release from soils requires microbial activities that isn't necessarily there in intensive farming application, and therefore farmers use uh, P from mining, and the deposits, global phosphorus deposits deplete. It has a severe environmental effects, and finally it increases the prices, so it has a severe economic effect on almond farming or in farming in general. And we, of course, suspected that optimal P applications could improve almond productivity because phosphorus supports physiology. So we set to determine the effects of phosphorus on tree physiology in summer. It enables metabolism. So we went to study how P availability enables carbohydrate metabolism of almonds, which are deciduous trees in winter. And finally, we wanted to see whether there is relations between phosphorus availability and what we call system services. So pollination by insects, and then fruit set and yields of almond trees in Israel. We used our unique setup. We have big lysimeters, actually huge. They're 1 cubic meter tanks where we grow trees for maturity and production. And we could monitor both the irrigation and drainage. In this case, we treated the trees with anything between 0 ppm phosphorus and a constant concentration or rate of 20 ppm phosphorus. We did this in 4 replicates, so 4 different trees, 4 different containers for each treatment, and a 4-year study. And we accompanied this system with periodic physiological and metabolic observations, and I will show you as we go on. So in this system, we always fertigate at a constant concentration. We determine whether there is drainage. If there is no drainage, then we fix the irrigation rate. It usually happens multiple times during the growing season, and from this we determine the evapotranspiration of these trees. We also collect drainage. Once there is drainage, we collect it, we quantify it, we analyze its mineral composition, and by this we can determine the multiple nutrient uptakes, and in this case, phosphorus uptake by almond trees. So first we learned that P deficiency affects almond trees, water, and phosphorus uptake. So almond trees can, you see here, they can actually on the left panel, you can see they could transpire more than 200 liters a day for a single tree. But if we see the yellow ribbons and the red ribbons, so in case of 3 ppmp or 1 ppmp, the trees actually transpired less than 100 liters for the better part of the growing season. It also affected their phosphorus uptake. So almond trees, in most common applications, extract about a single gram of phosphorus in a day. The more we applied phosphorus, the more they extracted nearly. So our system peaked at an uptake of 3 grams phosphorus a day for a tree. It actually, trees exposed to 20 grams, 20 ppms did not benefit from this further. And it meant that we could apply these 3 grams at a 75% efficiency, which is actually good considering that we still have a substantial leaching fraction, a 30% leaching fraction in this system. So actually trees won't necessarily see all the water and nutrients passing through the root zone. So moving on, we actually learned that besides these acute deficiencies, P did not affect the summer physiological processes of almond trees. So very low P levels reduced the photosynthetic rate. This is the red bars on the left here, and the chlorophyll content. This is the bluish bars on the left here. But then anything above 3 ppm P, all the trees photosynthesize the same. And looking at the right, we see that they also, the stomata conductance was actually pretty similar throughout the board, and also the corresponding stem water potential. So these were well watered trees. They never they sustained a constant stem water potential of anything between -15 and -17 bars throughout the growing season. This is actually low levels, but that's for another topic. So summing up the irrigation season, we concluded that high P applications won't necessarily benefit almond trees. Maximum LAI was yeah, leaf area index peaked in June. That's the red bar on the top, and it was similar for all the trees between 6 and 20 ppm P. Later, these differences actually even mellowed because the leaf area index was smaller since August. And same goes for stem radial growth, peaking in June for the 6 ppm treatment from all the other treatments, also decreasing for the rest of the growing season and showing little differences between the treatments above 3 ppm P. And going back to this plot at March to measure the actual canopy volume, it was about 20 cubic meters for all trees in the 6 ppm treatment and above. And the flowering intensity, so an visual index that we made, was also similar for all the trees receiving 3 ppm of phosphorus and above. But, and that's a big but, P actually promotes almond productivity. We measured this. So we measured bee visitation, and it turned out that fewer bees visit almond trees seeing less phosphorus. So about 2 bees visit an almond tree that sees 1 or 3 ppm phosphorus, and then 8 bees at the same time visit the neighboring trees. So this is a plot or a random design where just one tree stands next to the other in these bee containers. So there could be 2 trees, one receiving 15 ppm phosphorus and therefore 8 bees a minute, and the tree standing next to it, looking the same as you've seen from the summer observations, only had 2 visits or 2 bee visits a minute. That really affected the fruit set. So fruit set increased from about 8% in the low P applications to over 16% in the high P applications, and that affected also the yields. It essentially made it to 20 kilograms of dry kernel weight for a single for a single tree for the growing season. Essentially these values are actually low when we think of field production, but in case of the yields were about a single kilogram for a tree for this season. The trees were still young, and also in this experimental setup, there's other limiting factors for actual production. So moving on to see what would be the physiological mechanism that enabled this excessive production, but that we couldn't observe in summer, we found that phosphorus enabled carbohydrate metabolism. So looking at in December, at the time of senescence, the more phosphorus there was, the more phosphorus accumulated in almond branches. the more starch accumulated in both almond branches and almond roots. And then starch was depleted, trees depleted their starch reserve throughout winter, and by March it was these levels were much lower. But thing is, the depletion rate was higher for trees that had more access to phosphorus. So not in roots. This is the here on the right. You could see that in roots in blue, all the trees utilize the same amounts of starch, but in the branches, the phosphorus enabled a double or doubling the utilization of the starch reserves. And this translated to much more nectar. And so this is the panel on the left. What you see is the bars, that's the nectar volume. And then what you see in the red is actually the nectar sugar concentration. So the sugar concentration was similar for the flowers of all these trees, but the sugar volume changed from 2 microliters to 8 microliters per flower. That meant there was so much more sugars in these flowers. And this is what you see to the right, the amount of total sugars in a single flower actually correlated to the starch depletion that we observed in the branches. So we made a diagram and a manuscript, of course, that we published in Journal of Experimental Botany, where we explain how summer pea applications enable, of course, all the critical physiological apparatuses, but also accumulation of carbohydrates, starch, that then depletes, and it depletes more if there is more phosphorus. In winter, essentially converting into nectar and sugar in nectar that attracts more bees, and therefore there is more cross-pollination and more yields or almond yields. And this is an ongoing project or work. And what we need now to do is advance these pea applications in the field. So the first limit is the fact that we don't have mineral indices for these conditions. We have mineral indices. This is the purple ribbon on the upper panel of leaves pea concentration in July. It increases between 1 and even 6 or 10 ppm phosphorus, but later on we cannot observe any differences in phosphorus concentrations in leaves. When we came back and repeated this measurement in September, we couldn't identify any differences in phosphorus concentrations. Looking into the roots, seeing all these changes in carbohydrate metabolism in roots, we couldn't see any differences in phosphorus concentrations in roots in applications between 6 and 20 ppms of phosphorus. So we need to establish better indices that go beyond phosphorus deficiency. Then we need to gather more information by these indices on the actual conditions of phosphorus conditions of almond trees in field. And once we did that, we could suggest better applications, probably ones that follow the application rates that we recorded in our lysimeter study for the field conditions. Thank you. This work was funded by the CFPN and supported by the Israeli almond growers. And yeah, hope to see you in the questions session.